Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 50. The topic is an accounting of ATP production by cellular respiration. Discussing agenda for the topic will be an accounting of ATP production by cellular respiration. Lecture outcome. After watching and listening this lecture, students will be able to find out the total number of ATPs produced from the complete oxidation of a glucose molecule. Introduction. Now that we have studied the three major steps of aerobic cellular respiration, that is glycolysis, citric acid cycle, and electron transport chain. Now it's the time to calculate the total number of ATPs from this whole process. In the last few sections, we have looked rather closely to the key processes of cellular respiration. Now let's take a step back and remind ourselves of its overall function. Now the overall function of this whole process is to harvest the energy which is stored in glucose molecule and then to incorporate this energy in the synthesis of ATP. During respiration, most energy flows in this sequence. So first, the glucose molecule is broken down into two molecules of pyruvic acid, and in this process 2 NADH molecules and 2 net ATPs are produced. Then the pyruvate molecule is converted into acetyl coenzyme A, and because there are 2 pyruvate molecules, so 2 more NADH molecules are produced, and then acetyl coenzyme A enters into Krebs cycle where 3 NADH and 1 FADH2 is produced. Because the pyruvate molecules produced are 2 in number, so the Krebs cycle occurs twice. So all this NADH and FADH2 population then rushes to participate in the electron transport chain, and we have already discussed electron transport chain, which consists of a series of complexes embedded in the inner membrane of mitochondria, and as the electrons delivered by NADH and FADH2 passes through different electron carriers, energy is released. This energy is used to draw or to pump out protons or hydrogen ions into the intermembrane space, and as these protons present in the intermembrane space possesses potential energy, they are in bulk, and because of their concentration gradient they move back into the lumen again, courtesy of the ATP synthase channels, and in this way ATP is synthesized. So we can do some bookkeeping, so some record can be kept to calculate the ATP profit when cellular respiration oxidizes a molecule of glucose to 6 molecules of carbon dioxide. In Krebs cycle, 2 carbon dioxide molecules are produced. In link reaction, that is the conversion of pyruvate to acetyl coenzyme A, 1 carbon dioxide is produced. So each pyruvate molecule results in the production of 3 carbon molecules, and because 2 pyruvate molecules are produced from the breakdown of a single glucose molecule, so overall a total of 6 carbon dioxide molecules are produced, and all the 6 carbon molecules which are present in the glucose are broken down. The 3 main departments of the metabolic enterprise are glycolysis, pyruvate oxidation, and the citric acid cycle. This is the second department. The third department is the electron transport chain, which drives oxidative phosphorylation. Now this is the ATP yield per molecule of glucose at each stage of cellular respiration. So in glycolysis, 2 net ATP molecules are produced and 2 pyruvate molecules are produced, and along with that 2 NADH molecules are produced. Now remember that the process of glycolysis takes place in the cytosol, in the cytoplasm, whereas Krebs cycle and electron transport chain occurs in the mitochondria. So the NADH molecules which are produced in the cytosol must first enter into mitochondria, only then they will be able to deliver their electrons. The conversion of pyruvate to acetyl coenzyme A takes place in the mitochondria also. Then the acetyl coenzyme A, which is synthesized, enters into Krebs cycle. So the conversion of pyruvate to acetyl coenzyme A produces one NADH molecule, and because there are 2 pyruvate molecules and they are converted into 2 acetyl coenzyme A, so 2 NADH molecules are produced in this link reaction, and then these 2 acetyl coenzyme A enters into citric acid cycle. A single citric acid cycle produces 3 NADH and 1 FADH2. So 2 acetyl coenzyme A will produce 6 NADH and 2 FADH2 molecules. And the Krebs cycle also produces 1 GTP. So for 2 acetyl coenzyme A, there will be 2 ATP molecules. And remember, these 4 ATP molecules are produced through substrate level phosphorylation, when one of the substrates, one of the intermediates of the metabolic pathway, provide phosphate group 2 ADP molecules. And then in oxidative phosphorylation, the major bulk of ATPs are produced, which is 26 to 28. This figure is not exact. You can see here 26 or 28, 30 or 32. So why we don't have these exact figures? Because there are some things which are a bit doubtful. That is, these NADH molecules which are synthesized in the glycolysis, they must enter into mitochondria. Now the NADH molecule cannot travel directly and enters mitochondria. It must shuttle, it must deliver its electrons to another carrier, and once inside, these electrons will be combined either with NADH or FADH2. So once that happens, then it does have effect on the overall ATP production, because remember, NADH enters complex 1 and FADH2 enters complex 2. So the net combination, the net ATP production by NADH and FADH2 is different. And there is also one more thing which is a bit ambiguous, that how many hydrogen or protons should pass through the ATP synthase to synthesize one ATP molecule. And furthermore, once the ATPs are synthesized inside the mitochondria, they are transported to outside to the cytoplasm, where they are used as a source of energy. So it also requires energy to transport a ATP molecule to outside, and similarly the ADP molecules from the cytosol are also brought inside, so it also requires energy. So because of those reasons, the exact figure of ATP production from the complete oxidation of a single glucose molecule is not given. Now, figure 1 gives a detailed accounting of the ATP yield for each glucose molecule that is oxidized. The tally adds the 4 ATPs produced, 2 in glycolysis and then 2 in 2 citric acid cycles, and all the 4 are produced through substrate level phosphorylation and the citric acid cycle to the many more molecules of ATP generated by oxidative phosphorylation. So remember that the major chunk of ATPs is produced through oxidative phosphorylation in mitochondria. Now, each NADH that transfer a pair of electrons from glucose to the electron transport chain contributes enough to the protomotive force to generate a maximum of about 3 ATP molecules. Because the electron transport chain is not coupled with the synthesis of ATP molecules, when these electrons are passed from one carrier to another through the series of several oxidation reduction reactions, then these oxidation reduction reactions releases energy, and that energy is used to pump protons from the inner lumen into the intermembrane space, and then these protons from the intermembrane space return back to the lumen through ATP synthase, and in this process ATPs are synthesized. So these two processes, the transfer of electrons from one carrier to another carrier and the synthesis of ATP via ATP synthase, are two different processes. Now, why are the numbers in figure 1 inexact? In the last slide you saw that the values given were 26 to 28 or 30 to 32. So why these figures are not exact? There are some reasons for that. There are 3 reasons we cannot state any exact number of ATP molecules generated by the breakdown of one molecule of glucose. So we cannot put up an exact figure for the number of ATP molecules synthesized after the complete oxidation of a glucose molecule. There are 3 major reasons for that. The first reason is phosphorylation and the redox reactions are not directly coupled to each other. So phosphorylation is the synthesis of ATP molecules by ATP synthase. Redox reactions takes place in the inner lumen and they are not directly coupled to each other. So the ratio of the number of NADH molecules to the number of ATP molecules is not a whole number. So we cannot say that 10 NADH molecules will produce that number of ATP molecules, because the processes occurs in different compartments. Now we know that one NADH result in the pumping out of 10 hydrogen or 10 protons into the inner mitochondrial membrane. But the exact number of hydrogen ions that must re-enter the mitochondrial matrix via ATP synthase to generate one ATP has long been debated. So because the NADH molecules enter complex 1 and then its electrons moves all the way to the oxygen, which is the final acceptor of electrons. So this, in this series of oxidation reduction reactions, enough energy is generated to move 10 protons into the intermembrane space, and then how many protons must enter through ATP synthase to provide enough energy for the synthesis of one ATP molecule is something which is still under debate. Here once again we revise the whole process. These are the 4 complexes which are embedded in the inner membrane of mitochondria. So as the NADH molecules enters complex 1, they passes their electrons to a series of electron carriers, and the energy released to these oxidation reduction reactions helps in the pumping out of protons into the intermembrane space, and because the intermembrane space is having a high concentration of protons, and because of their abundant number and concentration gradient, they move into the ATP synthase, and in this way ATPs are synthesized. Remember, NADH enters complex 1, FADH2 enters complex 2. So based on experimental data, however, most biochemists now agree that the most accurate number is 4 hydrogen ions. So researchers are of the opinion that if 4 hydrogen ions passed through ATP synthase, then it produces enough energy to form an ATP molecule. Therefore, a single molecule of NADH, because NADH molecules provides enough energy to draw 10 protons into the intermembrane space, and 4 protons must sneak in through ATP synthase to produce 1 ATP. So a single NADH molecules provides enough energy to synthesize 2.5 ATP molecules. The citric acid cycle also supplies electrons to the electron transport chain via FADH2, but since FADH2 enters complex 2, so it only draws 6 protons into the intermembrane space, and as a result a single FADH2 molecule only helps in the synthesis of 1.5 ATP molecules. Now these numbers also take into account the slight energetic cost of moving the ATP formed in the mitochondria out into the cytosol, where it will be used, because ATP molecules are synthesized inside the mitochondria and they are transported outside into the cytosol where they are used. So this movement of ATP molecules to the cytosol also consumes some amount of energy. So these values are also taking into consideration the energy expenditure there. Now the second reason that why the number of ATPs produced from the oxidation of a single glucose molecule are not exact is that the ATP yield varies slightly depending on the type of shuttle used to transport electrons from the cytosol into the mitochondria. Now remember that the first stage of aerobic cellular respiration is glycolysis, where 2 NADH molecules are produced, but glycolysis takes place in the cytoplasm, so these 2 NADH molecules must be transported to the mitochondria to participate in the electron transport chain, but these NADH molecules cannot enter into mitochondria. They must deliver their electrons to another acceptor which is present inside the mitochondria. Now the NADH molecules can either deliver their electrons to another NAD molecule waiting inside or an FAD molecule waiting inside. So the mitochondrial inner membrane is not permeable to NADH, so NADH in the cytosol is segregated from the machinery of oxidative phosphorylation. The 2 electrons of NADH captured in glycolysis must be conveyed into the mitochondria by one of several electron shuttle systems. Depending on the kind of shuttle in a particular cell type, the electrons are passed either to NAD or FAD in the mitochondrial matrix. So if the electrons are passed to FAD, as in brain cells, only about 1.5 ATPs will be produced from these 2 NADH molecules which are present in cytosol. Whereas if the cytoplasmic NADH molecules deliver their electrons to NAD present inside the mitochondria, then the ATP production will be different. If the electrons are passed to mitochondrial NAD, as in the liver cells and heart cells, The yield is about 2.5 ATP per NADH molecule. Once again, revising it, glycolysis produces 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate. Remember, glycolysis takes place in the cytoplasm and electron transport chain, and Krebs cycle takes place in the mitochondria. So these NADH molecules must be transported inside into mitochondria so that they can deliver their electrons to the electron transport chain. However, it is not possible because the inner membrane is impermeable to NADH, so the NADH present in cytoplasm must deliver its electrons to an acceptor present inside mitochondria, which can be NAD and FAD. So when NADH delivers electrons to NADH, then maximum amount of ATPs are produced because they enter complex 1, and when NADH deliver their electrons to FAD, then 1.5 ATP molecules are produced because FAD enters complex 2. Similarly, the conversion of pyruvate to acetyl coenzyme A produces 2 NADH. The citric acid cycle, which occurs twice for 1 glucose molecule, produces 6 NADH and 2 FADH2 molecules, and then all these electron carriers arrives at the electron transport chain and deliver their electrons. So 2 ATPs produced through substrate level phosphorylation in glycolysis, 2 ATPs produced for 2 molecules of acetyl coenzyme A in Krebs cycle, and then the major chunk of ATPs are produced in mitochondria through oxidative phosphorylation. Now, a third variable that reduces the yield of ATP is the use of the proton motive force generated by the redox reactions of respiration to drive other kinds of work. For example, the proton motive force powers the mitochondria's uptake of pyruvate from the cytosol. However, if all the proton motive force generated by electron transport chain were used to drive ATP synthesis, 1 glucose molecules would generate a maximum of 28 ATP produced by oxidative phosphorylation plus 4 ATPs produced from substrate level phosphorylation to give a total yield of about 32 ATP molecules, or only about 30 ATPs if the less efficient shuttle were functioning. Less efficient shuttle means if the cytoplasmic NADH deliver the electrons to the FAD molecules present inside the lumen. Now, some of the energy generated by proton motive force is used to draw pyruvate into mitochondria. So this energy could be used towards the synthesis of ATPs, but is instead consumed in the movement of pyruvate to mitochondria. So this is yet another reason. Now, pyruvate molecule enters the mitochondria through a transport protein and is processed by a complex of several enzymes known as pyruvate dehydrogenase. Now, this complex catalyzes the 3 numbered steps: the conversion of pyruvate to acetyl coenzyme A. First, carbon dioxide is liberated. The remaining 2 carbon portion donates its 2 electrons to NAD and forms NADH, and then coenzyme A is combined with this 2 carbon portion and forms acetyl coenzyme A. The acetyl group of acetyl coenzyme A will enter the citric acid cycle. Then, now we can roughly estimate the efficiency of respiration. How efficient is the process of respiration? Efficient in a way that is this process efficient enough to derive all the energy which is stored in glucose molecules. So the percentage of chemical energy in glucose that has been transferred to ATP, recall that the complete oxidation of a molecule of glucose releases 686 kilocalories of energy under standard conditions. So this is the amount of energy which can be derived from the complete oxidation of glucose molecule under standard conditions. But what happens during this whole process? Phosphorylation of ADP to form ATP stores at least 7.3 kilocalories per mole of ATP. So this whole process is aimed to draw energy from glucose molecule and then store this energy into ATP. How much energy is stored by a single ATP? It is 7.3 kilocalories per mole. And how many ATPs are produced in this whole process? 32. So we multiply 7.3 with 32 and divide it by 686. So it will give us how much efficient this process is, how much energy was initially present in glucose molecule and how much energy was derived and stored in ATPs through this process. So the efficiency of this process is 0.34, and it is still a large value, because if you compare it with the energy that is converted by an automobile engine, is only 25%. That is, the energy stored in those fuels, only 25% of the energy is extracted, which helps the car to move. The rest of the 75% is liberated as heat. So the rest of energy stored in glucose is lost as heat. And remember one thing, that so many chemical reactions are involved and still the production of energy is only 0.34%. Now you can ask a question that what is the need for so many chemical reactions? The energy from glucose can also be drawn in a single step. So remember that in every energy transformation, some form of the energy is wasted as heat. So if all the energy in glucose is derived only in a single step, then a major chunk of this energy will be lost as heat. But now, because the process occurs gradually in so many steps, so the conditions are feasible for the cells to accumulate this energy, which is being produced slowly and gradually. Now we humans use some of this heat to maintain our relative high body temperature. The heat generated through aerobic cellular respiration is used for maintaining a proper body temperature, and some of this heat is dissipated to the environment. Now, surprisingly, perhaps it may be beneficial under certain conditions to reduce the efficiency of cellular respiration. Now, this is a very interesting statement, that the organisms, they thrive to yield maximum energy from any source of energy. However, there are certain cases in which an organism willingly tries to derive less energy. So, in which circumstances that takes place? A remarkable adaptation is shown by hibernating animals, the animals which undergo winter sleep, which overwinter in a state of inactivity and lowered metabolism. Although their internal body temperature is lower than normal, it still must be kept significantly higher than the external air temperature. One type of tissue called brown fat is made up of cells packed full of mitochondria. So the brown tissues has abundant amount of mitochondria. And now the inner mitochondrial membranes contain a channel protein called the uncoupling protein that allows protons to flow back down their concentration gradient without generating ATPs. Now recall the statement from the previous slide that some of the organisms willingly produce less amount of energy from the breakdown of glucose or other organic molecules, and therefore these organisms have another channel instead of ATP, which helps in the movement of protons into the mitochondria again without the production of energy. So why they do so? Activation of these proteins in hibernating mammals result in ongoing oxidation of stored fuel fats, generating heat without any ATP production. In the absence of such an adaptation, the buildup of ATP would eventually cause cellular respiration to be shut down by regulatory mechanisms that will be discussed later, because they doesn't requires abundant supply of ATPs. And remember what we studied in the regulation of these metabolic pathways is that when ATPs are present in abundant amount, then these ATP molecules can go back and they can attach with the phosphofructokinase enzyme and blocks its activity, so the glycolytic pathway is stopped. So therefore, to keep this glycolytic pathway running, they only produces a small number of ATPs and they keep on oxidizing the fat or the glucose molecules. Brown fat is also used for heat generation in humans. That's the end of the lecture. Thank you. Allah Hafiz.